Every neuron in the human brain is irreplaceable. Once lost, it never returns. Brain necrosis, a silent killer lurking within our skulls, marks the irreversible death of neural tissue. But what actually transpires in those critical moments when oxygen and energy vanish? How do toxins, infections, or trauma unleash microscopic chaos that cascades through our most vital organ? Today, we embark on a 3D clinical journey to uncover the hidden mechanisms behind brain necrosis, examine its root causes, and learn how early recognition can mean the difference between recovery and permanent damage. Brain necrosis represents the endpoint of diverse neurological insults, affecting millions worldwide through strokes, infections, and injuries. Despite cutting-edge imaging and molecular tools, once neurons die, they cannot recover. This reality demands that medical students and professionals understand every stage of necrosis, from its triggers to its cellular mechanisms, so they can act decisively when minutes count. Let's begin with defining brain necrosis and its biological nature. Brain necrosis refers to the uncontrolled death of neural cells marked by membrane breakdown, organelle swelling, and inflammatory signaling. Unlike apoptosis, which follows a precise and programmed sequence, necrosis is chaotic, destructive, and irreversible. Within necrotic tissue, ionic gradients collapse, intracellular contents spill into the extracellular space, and inflammatory cascades amplify cellular damage. In the brain, this process is devastating because neurons cannot divide or regenerate to replace lost cells. Surrounding tissue, acutely sensitive to biochemical disruption, succumbs rapidly as inflammatory mediators spread. Loss of vital neural circuits disrupts cognition, motor control, and autonomic functions. Ultimately, necrosis in the brain often leaves permanent deficits in memory, speech, movement, or may prove fatal. Next, we'll examine the various causes leading to brain necrosis. Ischemia is the most common trigger of brain necrosis. It begins when blood flow to neural tissue is reduced or blocked, depriving neurons of oxygen and glucose. Major clinical events include ischemic stroke, cardiac arrest, severe hypotension, or cerebral artery occlusion. Within minutes of oxygen deprivation, neurons exhaust ATP reserves, ion pumps fail, and calcium floods intracellular compartments. Activated enzymes such as proteases and lipases dismantle cell membranes and structural proteins. This rapid sequence, known as the ischemic cascade, culminates in membrane rupture, organelle swelling, and the uncontrolled death of neurons. Ischemic necrosis often progresses in distinct zones, with a central core of irreversible cell death surrounded by a penumbra of potentially salvageable tissue. Timely restoration of blood flow to this penumbra can rescue neurons and limit permanent damage. Next, infectious agents can directly invade or inflame neural tissue, leading to localized necrosis. Bacterial pathogens like Streptococcus pneumoniae or Staphylococcus aureus release toxins that injure cell membranes and provoke intense inflammation. Viral infections such as herpes simplex virus type 1 or West Nile virus can destroy neurons through cytopathic effects and immune-mediated damage. Parasitic organisms, including Naegleria fowleri and Toxoplasma gondii, penetrate brain tissue, causing rapid cell death. In each case, the release of microbial toxins and the host's inflammatory response drive a focal area of necrotic tissue and surrounding edema. Moving on, non-infectious injuries, including toxins, radiation, and trauma, can also precipitate brain necrosis. Carbon monoxide, heavy metals, methamphetamine, and industrial chemicals disrupt mitochondrial function, generate oxidative stress, and impair neurotransmission, driving neuronal death. In patients undergoing radiotherapy for brain tumors, delayed radiation necrosis can develop months or even years later, a consequence of vascular injury, blood-brain barrier breakdown, chronic inflammation, and glial cell damage. Traumatic brain injury, whether blunt force or penetrating trauma, produces hemorrhage, edema, and mechanical destruction of neurons, often compounded by secondary ischemia. These combined insults trigger mitochondrial failure, excessive free radical production, and a self-perpetuating cycle of inflammation. The result is progressive tissue loss and irreversible deficits in affected regions. As soon as necrosis begins, a biochemical storm takes hold inside neurons and their neighbors, setting off a deadly chain reaction that perpetuates damage long after the initial insult. Now let's delve into the pathophysiological cascades involved. Mitochondrial failure lies at the heart of necrotic injury. These organelles normally generate ATP, the energy currency that sustains neuronal function. When blood flow is disrupted or toxins accumulate, mitochondria become permeable, releasing reactive oxygen species and cytochrome C into the cytosol. Excess free radicals damage lipids, proteins, and DNA, accelerating cell death. Loss of ATP production halts vital ion pumps, destabilizing cellular homeostasis. The combination of energy failure and oxidative stress triggers rapid and irreversible necrosis in affected neurons. Calcium overload represents another lethal event. Under normal conditions, intracellular calcium concentrations remain tightly regulated. During ischemia or toxic insult, ion pump failure allows calcium to flood the cytoplasm. Elevated calcium activates proteases, phospholipases, and endonucleases, which degrade cell membranes, cytoskeletal structures, and nuclear material. The breakdown of these essential components leads to structural collapse and irreversible cell lysis. Calcium-mediated enzyme activation thus amplifies necrotic injury across neural tissue. The blood-brain barrier normally shields neural tissue from toxins and immune cells. In necrosis, endothelial tight junctions degrade, rendering the barrier permeable. Plasma proteins, inflammatory cells, and circulating toxins flood the brain parenchyma. This breach intensifies edema, elevates intracranial pressure, and accelerates cell death. Immune cell infiltration produces cytokines and free radicals, compounding oxidative stress. Breakdown of the barrier thus sets off a vicious cycle, deepening tissue damage and expanding the necrotic core. Microglia, the brain's resident immune cells, respond vigorously to necrotic debris. They release pro-inflammatory cytokines, nitric oxide, and reactive oxygen species. While intended to clear damaged tissue, this response often overshoots, perpetuating inflammation. Astrocytes and infiltrating macrophages compound the environment with additional inflammatory mediators. The sustained immune activation fuels oxidative injury, disrupts repair processes, and promotes further necrosis beyond the initial insult. This neuroinflammatory cascade is a hallmark of progressive brain necrosis. Moving on to the clinical symptoms of this condition. Before patients exhibit clear symptoms, subtle damage accumulates, quietly eroding neural networks until overt signs demand urgent medical attention. Clinical manifestations of brain necrosis vary depending on the location and extent of tissue loss. Patients may experience severe headache, often sudden and intense, signaling acute injury. Seizures can arise from hyperexcitable neurons in necrotic areas. Focal deficits such as paralysis, weakness, or sensory loss correspond to affected cortical regions. Memory impairment, speech difficulties, and behavioral changes reflect involvement of associative brain regions. In severe cases, loss of consciousness or coma may occur. When necrosis strikes the brainstem, vital functions like breathing and heart rate regulation can fail, often proving fatal. Additional signs include headaches, unresponsive to standard analgesics, subtle cognitive decline, and personality changes that may first appear as mild mood swings. Motor coordination and balance may deteriorate when cerebellar tissue is compromised. In some patients, laboratory findings reveal elevated inflammatory markers or abnormal metabolic byproducts in cerebrospinal fluid. Recognizing these early warning signs is crucial to prompt diagnosis and intervention. Then, we'll explore diagnostic tools used to identify necrotic damage. MRI, or magnetic resonance imaging, remains the gold standard for diagnosing brain necrosis. Its high resolution reveals areas of dead tissue surrounding edema and inflammatory changes. Diffusion-weighted sequences can distinguish acute necrosis from reversible injury. CT scanning provides rapid detection of hemorrhage and large necrotic cavities, a vital tool in emergency settings. While CT lacks the sensitivity of MRI, it remains indispensable when time or access is limited. Laboratory tests complement imaging by assessing systemic factors. Blood work can identify infection or metabolic disturbances, while cerebrospinal fluid analysis reveals inflammatory markers and potential pathogens. Toxicology screening detects neurotoxic substances that may underlie necrotic events. This multimodal diagnostic approach ensures accurate characterization of necrosis and guides targeted treatment planning. After that, we'll review treatment options tailored to each cause. For ischemic necrosis, restoring blood flow is critical. Thrombolytic drugs, administered within a narrow therapeutic window, dissolve clots and reopen occluded vessels. Mechanical thrombectomy, performed via endovascular techniques, can physically remove blockages in large cerebral arteries. Supplemental oxygen therapy supports tissue viability and reduces the extent of hypoxic injury. Early treatment of hypotension and cardiac events also helps preserve the ischemic penumbra and limit irreversible cell death. In cases of infectious necrosis, targeted antimicrobial therapy is essential. Bacterial infections require broad-spectrum antibiotics, later refined by culture and sensitivity results. Viral necrotizing encephalitis responds to antiviral agents such as acyclovir for herpes simplex virus. Parasitic causes demand antiparasitic drugs and, in some abscess cases, surgical drainage. Adjunctive corticosteroids can reduce inflammation and edema, decreasing intracranial pressure and protecting surrounding neural tissue. For toxic necrosis, identifying and eliminating the offending agent is the first priority. Carbon monoxide poisoning demands immediate administration of high-flow oxygen or hyperbaric oxygen therapy. Heavy metal exposure may require chelation therapy to bind and remove metals like lead or mercury. Supportive care, including intravenous fluids and antioxidants, helps mitigate oxidative stress. Continuous monitoring in an intensive care setting ensures hemodynamic stability and organ support. Radiation-induced necrosis requires a different approach. High-dose corticosteroids reduce vascular permeability and control perilesional edema. Hyperbaric oxygen therapy may enhance tissue oxygenation and promote healing of irradiated areas. Anti-inflammatory medications, such as bevacizumab, can target vascular endothelial growth factor to stabilize blood vessel integrity. In refractory cases, surgical resection of necrotic tissue or stereotactic radiosurgery for residual tumor must be considered. Management of traumatic brain injury focuses on preventing secondary injury and controlling intracranial pressure. Surgical interventions, such as decompressive craniectomy, alleviate mass effect from hemorrhage or swelling. Intracranial pressure monitors guide osmotic therapy and sedation to maintain safe intracranial dynamics. Intensive care support, including ventilation, hemodynamic stabilization, and seizure prophylaxis, is essential. Rehabilitation services begin early to maximize functional recovery after the acute necrotic injury. Following treatment strategies, we'll discuss preventive measures. Prevention of brain necrosis centers on risk management and early intervention. Strict control of blood pressure, diabetes, and cardiac health reduces ischemic events. Protecting against infections through vaccination, prompt antimicrobial treatment, and safe hygiene practices limits infectious necrosis. Minimizing exposure to neurotoxins, employing protective headgear in sports, and monitoring radiation doses during therapy further guard against tissue injury. Early identification and management of head trauma, alongside rapid neuroimaging assessment, can avert progressive necrotic damage. Brain necrosis remains a formidable challenge because the loss of neural tissue is irreversible. Its complexity arises from the interplay of ischemia, infection, toxins, radiation, and trauma, each converging on mitochondrial failure, calcium overload, and barrier breakdown. Yet, understanding these mechanisms empowers clinicians to intervene strategically. Early recognition through advanced imaging and laboratory testing enables targeted therapies that can salvage vulnerable tissue. As we deepen our knowledge of neuroinflammation and repair pathways, future research holds promise for neuroprotective and regenerative treatments. The responsibility lies with medical professionals to identify necrosis promptly and apply evidence-based interventions to mitigate lifelong deficits. We stand at the threshold of breakthroughs in neuroprotection and cell therapy. Gene editing, stem cell transplantation, and novel anti-inflammatory agents are under investigation to halt or even reverse necrotic injury. As emerging clinicians and researchers, your role in advancing these therapies is vital. Collaboration across neuroscience, imaging, and critical care will